Okay guys, this is lecture 8, Movement, Muscles and Skeletons on Motors, Batteries and Locomotions. And for the recording I'm not going to cover all of the lecture, I'm just going to cover the most important bits that you might or may need to know for the exam. That doesn't mean it will all be in the exam, and that doesn't mean there will be exam questions on it, but I would advise you to listen to the bits I do cover and revise them. Alright. So in this lecture we would cover how simple organisms can move, amoeboid, flagellate and ciliate. We then look at muscle types, understand how muscle works, and then look at the difference between insect and crustacean muscle, and then look at skeleton types. But the most important thing we're interested in at the moment is how can simple organisms move, whether they use amoeboid, flagellate or ciliate movement. So looking at the learning objectives, what we're most interested in are how organisms move, and when you have time I'd suggest you have a look at the other things as well, because you might actually find them of interest. Right, these are the relevant chapters in the textbook. Okay, so thinking of just about very small microscopic organisms, before you move on to the next slide, there are 3 different ways that I'm going to say that simple microscopic organisms can move. So remember, for them their Reynolds number is high, so they're living in a very viscous environment. How do these small, simple organisms move? Alright, the 3 ways in which they can move is through amoeboid movement, so like amoeba, through flagellate movement, so using a long whip-like tail, or through ciliate movement. So flagellate things can have several flagellates, not limited to one, but they're long and whip-like. Ciliate movement, you normally have a series of cilia surrounding the outside of the cell. Okay, the first form of movement we're going to look at is amoeboid movement. And this is named, funnily enough, after amoeba. And when you look at the 3 videos here you want to have a look at, and when you look at the 2 amoeba videos, what you'll see is, what they do is they'll extend out a foot, or it's known as a pseudopodium or false foot. This foot extends out and then the rest of the amoeba appears to flow in behind it. So what happens is they can change the nature of their extracellular membrane and intracellular fluid, or basically they're changing the nature of their structure, so that it can change its viscosity, so that it allows its foot to relax, changing its viscosity. That flows outwards. Okay, the cytoplasm within inside or endoplasm then begins to flow into it. That begins to stiffen up the outside, so the whole organism moves forward. Now, in reality, what they're doing is, it's not like this magic fluid that can stiffen or soften. What they're doing is they've got microfilaments or a cytoskeleton inside, okay, which are driven by myosin motors, and we'll talk about that in a minute. Now, it's not just amoeba who use this amoeboid form of movement. Slime molds, which we used to think of fungi, but we now classify as protista. Mammalian leukocytes, as you'll see in the chloroplasts, in the cyclosis, they all use amoeboid movement in order to attain locomotion. Alright, this is a much better explanation, or a much better visualization of how amoeboid motion works. Alright, this is in lamellipodia, which are in epithelial cells and fibroblasts, so they're only 2-dimensional, unlike pseudopodia, but the principle is effectively the same. So what you can see is we have a series of proteins. Okay, we have an actin meshwork. Okay, and so the actin filaments are effectively the cytoskeleton and the bit of the muscles work against. Okay, and then we have a series of dynein motors. Alright, so what happens is the actin filaments disassociate at the back. Okay, diffuse molecules of actin flow forwards, bind on, okay, to the existing actin network and to the uncapped regions, and basically grow outwards. So what you have is you have the rearmost portion of the network disassociating and then reforming at the front, so the whole system actually seems to move forward. Right, you're probably going, my god, this is so complicated. You just need to understand the idea, and it'll probably become more clear on the next slide. Alright, so what we have here is we have actin and myosin. Okay, so actin are our cytoskeleton filaments, or our muscle filaments, and normally in humans then we have a myosin filament as well, but here, in our muscles anyway, here we have myosin particles actually bound to a bit of glass slide. The diagram below just shows you how the arrangement of the actin and myosin filaments, and what will happen is the actin will bond to the myosin will do a power stroke and move the actin filament along. On the top slide, what you can see is we've got 2 actin filaments, we've labelled in two different colours, one green and one red, and what we've done is we've provided the series with ATP, so that provides the energy for the myosin to do its work, and you can see that the myosin is actually actively shunting the actin filaments along them, so the filaments are in time-lapse are moving past each other. And that's exactly the same principle in a way how the lamellipodia work. Yes, they're being pushed along, and yes, the actin filament's growing at the new end, so you get this growing going along. But again, what we're seeing is we're seeing the pushing forwards, or this amoeboid movement. Alright, so that's amoeboid movement, but what about ciliate movement? So cilia, and as we'll talk about in a minute, flagella are molecular makers built from microtubules and dynein. Okay, so dynein is effectively a precursor of myosin. And if we look at our 92 arrangement of filaments here, okay, what we can see is we've got our central pair of two microtubules surrounded by our 9 pairs of outside or external microtubules. And on our microtubules, which we can think as actin filaments, okay, what we have is we have these dynein. Alright, now cilia are going to move backwards and forwards, so what I want to ask you is how are the cilia going to move backwards and forwards? Well, the answer is if you can contract on one side, then if you contract on one side, that will cause the filament to bend. The other side says relax, you then contract the other side, relax the other side, and this way, by alternately contracting and relaxing sides, you can get movement. Okay, you can get like a whip-like movement. And if you look down into the bottom left-hand side, what you can see is you can actually see a dynein molecule, and it's clearer on the next slide. And what you can see is that this tiny little molecule, when it's activated with ATP, changed its conformational shape from one position to another. So we've got an actual tiny pyrostroke, which is about 6 to 9 nanometers. Alright, and this is also on, which is shown as 8 nanometers on the following slide, which is a tiny amount of a tiny amount of movement. However, if you have loads of these and lots of movement, then you can generate activity or significant movement for a large molecule, or in this case the cilia. So here you can see our dynein attached to our microtubule, okay, or this could be myosin and actin in a vertebrate muscle. You can see that, okay, as ATP is phosphorylated to ADP plus a phosphate, okay, we have a change in conformational shape of the dynein molecule, so it goes from the right hand side to the left hand side through a distance, it says here a power stroke of about 8 nanometers, okay, and that pulls the microtubule along. Alright, for the cilia or flagella, okay, that pulling will result in contraction of one side or the other, depending which side or which microtubules you've activated relevant to the central axis and its other side. Okay, cilia are common in almost all organisms, okay, and it's always this universal 92 arrangement. However, when we talk about flagella in a minute, flagella aren't found in all organisms, or only found in certain life stages or certain specific cells in organisms. So, cilia and flagella both need aqueous environments in order to work, and I want you to think about why and answer that for yourself. When used for locomotion, microscopic organisms have the whole epithelia covered in cilia. However, in us, we have cilia in many places, and we obviously don't use it to move. So, for example, we have in our lungs, we have ciliated epithelium, which is to move fluid and particles trapped in our lungs out of our lungs to get rid of. And also, women have them in their fallopian, in their infundibulum, at the opening of their fallopian tubes, to generate a gentle current to guide any oocytes oocytes that are produced down into the fallopian tube so it can be fertilized. Okay, on the last slide there are two videos you can watch to look at ciliated locomotion. And here's a picture of a ciliated protozoa, and what we've got is an electron micrograph on the right hand side showing a cross section of the flagella of protozoa, and all we're interested in is if you count up the microtubules we can see the clear 92 arrangement. Okay, and as I mentioned, if we look in higher order organisms, we can that cilia are involved, and you should be able to tell me from the last lecture in invertebrate nephridial organs. Okay, so that's to generate the current pressure for filtration. We've seen them in bivalve gills, where we've talked about them using for feeding. I've mentioned them in lung epithelia, and I've mentioned them in fallopian tube epithelia in the infulbuldineum, which I can never pronounce. Okay, now these are flagella. They are different from cilia. Now they tend to be, organisms tend to only have a few flagella rather than many, and flagella tend to be a lot longer than cilia. So we normally talk about single or biflagella organisms, and their long whip-like tails that undulate. If you think about cilia, they just sort of like beat in like little strokes along the edge of an organism, whereas flagella tend to be directional. There's a couple of videos you can watch here. The only flagella that we as mammals have are in male sperm. Everything else is ciliated. There's no other flagella anywhere else in us. Now, normally when we think about flagella, we think that as the flagella beats, okay, it propels them in the opposite direction. However, just note that there can be backwards, forwards swinging flagellates where the actual flagella seems to pull them rather than push them away from the direction of beat of the flagella. In this case, the flagella isn't smooth, it's covered with like lots of little spines or spikes, and what's happening in this case is that the flagella, because of the Reynolds number of the organisms, because of their small size, they're living in a very viscous medium, it's effectively pulling them through the fluid rather than pushing them. Now, when I talked about cilia versus flagella, I said that cilia effectively just beat on the side of an organism, whereas the flagella whips in a tail-like motion. And what you can see here in figure 10.2 is the beat of a flagellum on the left-hand side, okay, and the beat of a cilia on the right-hand side. So what you can see is that the cilia on the right-hand side is just beating backwards and forwards, okay. So it's just basically moving in a stroke from parallel to the body, up through 90 degrees, back down through 180 degrees, parallel again to the body. In contrast, when we look at the flagella, okay, the flagella is beating perpendicular to the body. So when we look at water flow with a cilia, water flow in a ciliated epithelium is parallel to the surface. However, when we look at water flow from flagella, okay, it's perpendicular. So, ignoring backwards forwards flagella, okay, an organism, if it's got a flagella, by moving the flagella, will move in the opposite direction to the way the flagella is beating. However, with a cilia, okay, if an organism is just using one side of its ciliated epithelium, it will move parallel to the beat of its cilia. Alright, just to finish off now, just a quandary, and if you think about it, okay, and you think about Reynolds numbers, and you think about the force that cilia or flagella can develop, and we have talked about in lectures, why don't whales use flagella or cilia instead of flukes? So instead of like a whale having a big massive muscular tail, why doesn't it just coat its body in loads of cilia, or have just a single thin flagella to move it through its water column? Alright, that's all you really need to know for the final exam from this lecture, but I would advise you, when you get a chance, just to have a look through the rest of the lecture. In particular, you might be interested to know why, okay, zombies, it takes 24 or 48 hours after you die from being bitten by a zombie to actually turn into a zombie. This instantaneous zombies you see on TV nowadays, no, it's not true, and there's a good physiological reason why for it. Alright, that's it.